In this video, we're going to talk about the first and second part of plant diversity. The first part will be over green algae. There are three lines of green algae: colonial algae, tetrasporin algae, and siphonous algae. The examples that are provided in your lab manual include colonial and siphonous algae. I did not see any tetrasporin algae in your lab manual. We will only talk about the algae that are provided as examples in your lab manual. As we talked about last week, algae are protists. However, they fall into the supergroup of Plantae, so they are closely related to plants. They have plant-like characteristics. Colonial algae are basically single-celled algae that work together as one unit. So you're going to have a lot of different cells, single-celled algae, coming together and forming a group and functioning and working together as a group. There are several examples of colonial algae. However, the example that is provided in your lab manual is the Volvox. This is a hollow sphere containing 50,000 or more species of biflagellate cells that are equally spaced out in the sphere. As you see here, all the little dots that you see on the sphere, these are single-celled algae. And the combined beating of their flagellae causes the colony to glide and roll through the water. So they're basically synchronized. They work together and cause this Volvox to swim and glide through the water. Volvox can produce both sexually and asexually. If we take a look at a Volvox here, you'll see that it is going to be filled with these single-celled algae on the surface. If you take one of them and you blow it up, this is sort of what it looks like. In lab, we're not going to talk about the details of the organelles and everything like that. But as you can see, it is a eukaryotic organism, and it does have flagella, 2 of them to be specific, that help move the Volvox. Inside this colony, you're going to see these circles, these spheres. These are basically the daughter colonies. This is basically a form of asexual reproduction. These are little colonies that are growing inside of this big colony. And when these daughter colonies are ready, this colony here is going to rupture and release these daughter colonies. So here is an example of how a Volvox moves in the water. So the Volvox will reproduce asexually through the formation of autocolonies. We could see here's a colony here. These autocolonies, these are the daughter colonies right here inside of this parent colony. Not all cells can give rise to daughter colonies. Just like we have somatic cells in our body, and we also have our gametes, our eggs and our sperm cells, there are specialized cells here on this parent colony called the gonidium cells. And the gonidium cells are able to divide and form daughter colonies that are going to be small versions of the parent colony. The only difference here with these daughter colonies is that the flagella of the cells is going to be on the inside for a while it is growing in the parent colony. When the cells of the daughter colony divide into the same number of cells as the parent colony, so basically when they're large enough and there's enough cells on that daughter colony, the flagella is going to become inverted. It's going to invert and it is going to face outwards. So remember, first the flagella is going to be facing inwards. Once the daughter colony has grown enough, the flagella will face outwards. And eventually, as this as the daughter colonies grow, the parent colony will split open and release the newly formed autocolonies. So this shows the asexual reproduction cycle of a Volvox. We can start over here. We have one Volvox here. Inside of it, you can see the daughter colonies. They're small, and of course the Volvox is small. It is growing. And what's going to happen is that these cells are going to grow. And as you can see here, the daughter colonies are growing. Their number of cells will continue to increase. And when it has reached a certain number, the flagella of the cells of these daughter colonies will become inverted, so they will start to face outwards, and eventually this parent colony is going to split open and release these daughter colonies. And again, this cycle will just continue. Based on experiments done in lab, Volvox tend to prefer asexual reproduction. However, sexual reproduction is also important for their survival, especially when environmental factors are not very favorable, such as when there's heat or drought. These colonies are either male or female. So the male colonies will release sperm pockets that can fertilize a female colony. So this was the asexual cycle that I just talked about earlier, and this one over here is the sexual cycle. And we could see the sperm packets that are released here. The sperm packet will fertilize an egg inside a female colony, and then you're going to have something called zygospore, which is diploid, goes through meiosis, and then it's going to release this young Volvox over here. So again, Volvox can go through asexual reproduction, where it just like makes copies of itself inside of the sphere and releases them when they're ready, or it can go through sexual reproduction as well, because these Volvox are either going to be male or female. Next, we're going to talk about the siphonous green algae. These are going to be filamentous, unbranched algae. There are several different examples in your lab manual, and we will just talk about those specific examples. There's different ways to pronounce this. The British way is Eulothrix, and the American way is to pronounce it as Lothrix. These are going to be non-branching filamentous algae. You're going to find them in fresh and marine water. They prefer low temperatures, such as temperatures in winter or spring. Of course, they're eukaryotic. These are going to be multicellular algae. They tend to attach to surfaces, and they can go through reproduction both sexually and asexually. So in asexual reproduction, the contents of the cell are going to kind of condense like this and become something called zoospore. And then the zoospore can be released from the cell. If it falls onto a surface that is suitable for growth, it will grow a colony. Lothrix can also go through sexual reproduction, and this is when the vegetative cells are going to round up their content to become gametes. These gametes are going to be biflagellate, and they're going to be released from an opening of the cell walls. And then when they're released, they can pair up and form this quadriflagellate zygote that will develop into a resting cell. The zygote can actually last for a while. When the temperature is right for the growth of the zygote, it will then germinate. Next algae that we want to talk about is the Odogonium. There are several different types of Odogonium. You're going to find them in freshwater ecosystems. Again, these are unbranched filamentous algae. They're multicellular. They form patches on the surface of water, and they're going to include both sexual and asexual reproduction. And they have been found to be important in fixation of heavy metals in freshwater ecosystems. There are two groups of Odogonium. The first group is Macandrus species, and there's two forms of Macandrus species. We'll talk about them. There's the monoecious and the dioecious, and then there's the Nanandrus species. And I am not a plant expert, so I'm doing my best to pronounce these correctly, but I'm not 100% sure if I'm pronouncing them correctly. So first, let's look at the Macandrus species. The thing to keep in mind about these species, Macandrus species, is that the filaments are going to be normal size. Both types are going to have filaments of normal size. So they're going to have male sex organs and female sex organs. If you have a Macandrus monoecious species, the Antheridia and the Oogonia are always found on the same filament. Antheridia is the male sex organ and Oogonia is the female sex organ. So here's the Oogonium right here, and here's the Antheridia. So this is the male sex organ and this is the female sex organ. If it's monoecious, it's going to be on the same filament. Now you could have Macandrus dioecious. The Antheridia and the Oogonia are going to be found on different filaments. So this is a different species over here. Another species of Odogonium is the Nanandrus species, and the difference here is the filaments. What you're going to find here is that the filaments for the male organs are going to be different. So the filaments producing Antheridia and Oogonia are going to be morphologically different. Antheridia are going to be smaller than the Oogonia, and they look unique. They look like you can see it right here. They're called dwarf males. So this, of course, looks different than the Antheridia you saw here with the Macandrus species. So again, this is called the Nanandrus species, and it contains the dwarf males. Next algae we want to look at, this is the Spirogyra. The name, of course, is because of the way that the chloroplasts are going to be arranged inside of these filaments. They form this spiral shape, and that's why it's called Spirogyra. This is going to be an unbranched filamentous green algae. It is found in freshwater habitats and is often observed as slimy patches on top of water. So if you look at it on top of water, it looks something like this. You look at the filaments under the microscope, looks something like this. You could see these green spirals in the filaments. It can reproduce in several different ways. It can reproduce through the vegetative method, which means that by fragmentation, if there's pieces of it falling off, the pieces can grow. There's the asexual method, and this is by spore formation. They can produce spores, and then the spores can vegetate when the conditions are right. This is not the most preferred way of reproduction for Spirogyra. It goes through asexual reproduction if the conditions are very unfavorable. And then you have sexual method, which is it produces sexually, but in quite a unique way. There's different ways of sexual reproduction with this algae. There's something called the scalariform conjugation. Scalariform means ladder, so it kind of forms these structures that look like ladders. And there's lateral conjugation, which can come in two different forms, indirect and direct. So the only method that I noticed that I saw in your textbook, or not your textbook, your lab manual, is the scalariform conjugation. So that's the only one that I'm going to talk about. However, I did provide a link to a really good video that goes through all these different methods of reproduction for Spirogyra. So with the scalariform conjugation, and you see pictures of this in your lab manual, you're going to find two filaments that are going to be lateral to one another. So here's one filament over here, and here's another filament over here. Because they're morphologically pretty much the same, we cannot label the filaments as male or female. Even though they're physiologically different, the morphology is pretty much the same. So instead of calling the filaments male or female, we use the plus and the minus sign. So we could say that this is plus, the other one is minus. When these two filaments are close to each other, they're going to start to produce these protuberances, as you see here. The protuberances will eventually come together, as you can see here, touching each other, and the cell wall will disappear, and they're going to form this canal over here. So we could see the protuberances, when they join, they form these canals. And these canals are called conjugation canals. So the protoplast in each cell is going to condense. So this is pretty much the content of the cell, and it is going to become a gamete. So one gamete, the gamete of one cell, is going to migrate into another cell. The male gamete, which is in this case on the left side, is going to migrate through the canal into the female gamete and fertilize this egg. So we could see the movement happening here. This is called amoeboid movement, and it's going to come over here into the female gametes over here, or the eggs, and it's going to fertilize the egg. And here you're going to have a diploid zygote that is going to be formed. Again, the gametes are going to be morphologically pretty much identical, but physiologically different. So I shouldn't really use the term sperm or egg just because they are morphologically identical. So female gametes and male gametes would be more appropriate terms to use. Now this is not the only way. There's lateral conjugation where you have like side-by-side cells doing this fertilization. And there are some cases where this fertilization can happen in the canal. I'm not going to go over those because those are not in your lab manual. This is the only one I saw in your lab manual, so this is really the one that you're responsible to know for your quizzes and your exams in lab. However, if you want to review the other methods of reproduction, there is this great YouTube video. It's about 7 minutes long, and it goes through every method of reproduction in Spirogyra. Okay, now let's go over part 2 of plant diversity. So we're moving on from the algae, and we're going to go to the lower plants. So these are pretty much the first land plants that have evolved. The first ones are going to be bryophytes or bryophyta. These are non-vascular plants. So these are plants that you see on the ground or growing on surfaces of things, but they don't become tall because they lack a vascular system. So they're limited in size. They prefer moist habitats, and we will see why, because water is going to be very important for reproduction of these plants. They produce enclosed reproductive structures. Most of them, as we'll talk about, can produce sexually and asexually. So they're going to have these reproductive organs called gametangia. Gametangia produce gametes, and these are going to be important for sexual reproduction. And sporangia are going to produce spores, and those are going to be important for asexual reproduction. These plants don't produce flowers or seeds, and as we talked about, they produce sexually and asexually. They are quite important for our environment. They are photosynthetic, so they produce oxygen. They hold a lot of water. So for example, moss holds a lot of water. So they lower humidity in the environment just a little bit, and they form symbiotic relationships with other plants. Here are some examples of bryophytes. This is one that we're going to talk about in a little bit of detail. These are liverworts, these are hornworts, and these are mosses, and these are all considered to be bryophytes. One of the examples you're going to have to look at in lab and look at slides and draw are liverworts. So these are going to be land plants, of course. They don't have a vascular system. They don't produce seeds, but they do produce spores. They are required to be in wet or moist environments. And they don't have true leaves, they don't have true stems, they don't have true roots. So these are not really leaves. So we sometimes use the term like leaf-like structure or stem-like structure or root-like structure. There's different terms for these, as we will see. So the liverwort that you're going to look at in lab is the Marchantia. And there's quite a number of slides from Marchantia in your lab manual. So do look at this. Make sure you're looking at this in lab. You're supposed to draw them and include them in your lab report. And of course, a good shortcut would be to use your cell phone to take pictures, and then you could go back and draw them later. But just keep in mind that you're supposed to draw them and not just include pictures from your phone. With the Marchantia, you're going to find these leaf-like structures over here. Let me go back. These leaf-like structures. They're not called leaf. They're called thallus. In liverworts, they're called thallus. So this is a thallus over here, and this is a thallus. The thallus can be male or female. So this one is a male thallus, and this one is a female thallus. They look almost identical, except for these structures that are growing on top of them. This is called an archegoniophore. This is going to be sort of like the female structure. It's going to contain the eggs. And this is the antheridiophore. This is going to contain the sperm. So this is a male structure. And then you're going to see the structures on top of the thallus. They're sort of like cups. These are called gemma cups. We'll talk about them in a little bit. The gemma cups are important for asexual reproduction. So these structures here are important for sexual reproduction. And these little cups here are important for asexual reproduction. And then if you look underneath the thallus, you're going to see these root-like structures called rhizoids. We don't call them roots, but they pretty much serve the same type of function. So instead of calling them roots, we call them rhizoids. So one of the slides you're going to look at is the thallus of the Marchantia. You're going to see two sides that has two portions, two surfaces, I should say. There's going to be this upper photosynthetic layer. You're going to see these photosynthetic filaments here. And then there's going to be this lower storage layer over here. Some things to pay attention to are, for example, this upper epidermis layer. And within this upper epidermis layer, you find that there's going to be these tiny little openings. They're called air pores, and this is where gas exchange takes place. You're going to find photosynthetic filaments here, right here. And then there's going to be something called the scale over here. The scale is a structure that protects the growing of the thallus. And the rhizoid over here. And the rhizoid facilitates the absorption of minerals and water from the soil. There is a picture of this in your lab manual, and you will look at it in lab. Again, we talked about a male and a female thallus. So let's talk about it in a little bit more detail. The male thallus, again, this is a thallus, is going to have something called the antheridiophore. And here is the antheridiophore over here. It's like sticking up from it. It's going to have the male sex organs. And what you'll notice with the antheridiophore is that when you look at it, the top structure here, it is disc shaped. And it's actually very important for it to be disc shaped. And you'll see why. This is important for fertilization. And the antheridiophore is going to contain these organs called antheridia, and the antheridia are going to have the male gametes. So if we take this and look at it under the microscope, you're going to see something like this over here. And you see these structures here. These are the antheridia, and they're going to contain the male gametes. This is what the antheridiophore look like. They're flat and disc-shaped on top. Now let's look at a female thallus. With the female thallus, everything's almost identical except this structure over here. This is called the archegoniophore, and it's going to contain the female sex organs. When you look at the archegoniophore, you're going to notice that it's not going to be disc shape. It's going to have these finger-like structures. Kind of looks like an umbrella, or and it's not going to be disc shaped. And it's going to hold the egg-producing archegonium. So within the archegonium, right here, you could see the egg. So the sperm is going to have to travel down this tube and come over here to fertilize the egg. And when you look at it, the archegoniophore looks like this. Sort of umbrella shape, sort of has these finger-like projections. So now let's talk about how sexual reproduction takes place in Marchantia. Water is going to be required for sexual reproduction, and that's why liverworts really only grow in moist environments, in environments where there's a lot of water. So water is going to be the transportation mechanism here. So when water, let's say it rains, the water is going to get splashed on top of the disc-shaped antheridiophore. And remember, the antheridiophore is going to have the male organs. So water is going to initiate sperm release from the antheridia when water is splashed on it. When water splashes on the antheridiophore, it's going to basically help the sperm travel through drops of water and fall on the female organ. So the sperm are going to be transported from drops of water to splash from the flat platform of the male. So this disc structure is important for splashing of water. The sperm then reach the opening of the archegonium and swim down a protective canal to reach the single egg. And then once it reaches the egg, fertilization can occur, and the embryo will grow to form a diploid sporophyte. And of course, from there, the spores can travel if they fall on the... So here is the female, here's the male, and let's say it rains, you're going to have these sperm gametes being released. They're going to fall on top of the female, and here you could see that the egg, if it travels down this tube, the egg is going to become fertilized. It becomes an embryo and a spore, and the spore can then fall into the ground on a soil, and it will germinate, develop rhizoid, and it will become a liverwort. So this is sexual reproduction. Now, asexual reproduction can also happen if you pay attention right here to these cup-like structures on top of the thallus. These are called gemma cups or gemma cups. And when water fills these cups, let's say through rain, and water fills them, you're going to have these cells that are called gemma that are going to float out of these cups, fall onto the soil, they're going to germinate, and they're going to produce more Marchantia. So this is how asexual reproduction takes place with Marchantia. Here is a short video of the cycle of liverwort, and it's quite short. I think it's less than 2 minutes if I remember correctly, but it is a pretty good video. I recommend that you watch it. Okay, now let's move on to some vascular plants. In your lab manual, it states that you're supposed to pick one of these slides of these plants and look at them under the microscope and draw them. Again, you don't need to look at every single one of them. They ask you to pick one of them. So you could pick Lycopodium, Selaginella, or you could pick Equisetum. Now, before we end this video, we want to talk about ferns a little bit. Ferns are, again, one of the first plants that evolved on our planet. However, they came after these non-vascular plants, such as mosses, liverworts, and hornworts. They are vascular, so they tend to get taller. They don't have leaves, so we still say leaf-like structures. The leaf-like structures on ferns are called megaphyls. These are one of the oldest plants, of course. They don't have seeds, so these are seedless plants. Just like mosses, liverworts, and hornworts, they're seedless and they don't produce flowers. If you take one of these megaphylls that belong to ferns and you look at it, look underside of the megaphyll, you're going to notice that there's these black or brown dots. These are called sori, and they're going to contain a group of sporangia in them. Now remember, sporangia are where asexual spores are going to be produced. These ferns can be divided into two groups based on the structure of their sporangia. There's eusporangia or eusporangiate or leptosporangiate. These are the two different types. So here you could see a eusporangiate over here. And here, if you look at it under the microscope, it looks like this. What you'll notice with the eusporangiate is that the sporangial wall has several layers, so it is going to be relatively thick. It is going to contain a large number of spores, and it has a thick stalk over here. This is, I believe, pronounced Bothridium sporangium. And the other type is going to be called leptosporangiate. This is going to have a single layered wall around it, so it's not going to be very thick. It's going to have a limited number of spores, and it is going to have a slender stalk. And there's different examples of leptosporangiate in your lab manual that you could look at. Now, reproduction of ferns is not in your lab manual, but I want to talk about it very briefly. We're not going to go into details just because it's not included in your lab manual. Fern can be produced by asexual reproduction or sexual reproduction. Asexual reproduction can happen when there's propagation of a new plant from rhizomes in the soil. So from these root-like structures that are in the soil, you could grow more and more of them. And that is why some species of fern can really grow uncontrollably. And then sexual reproduction happens from the spores. So, and it goes through two phases, this like sporophyte phase and this gametophyte phase. So we talked about these megaphils containing those little structures underneath them called sori, like this. And what's going to happen is that from here, spores are going to be released. When spores are released onto a soil, they're going to germinate, and they're going to give rise to gametophytes. And the ferns are able to produce on a gametophyte both the male heads and the female heads. So when there's things such as rain, the male gametes will travel to the female gamete, and they will cause fertilization of the egg. And when this happens, a new plant will develop, a new fern will develop. So this is the cycle of sexual reproduction in fern. And again, it could also develop asexually. Okay, this is it for plant diversity part 1 and part 2. And for the next video, we will probably do plants that are vascular, but also have seeds and produce flowers.